Plasma membrane. Plasma membrane is also called cell membrane. It is the boundary of protoplasm and most organelles. It is found in all living prokaryotic and eukaryotic cells. It controls the passage of material into and out of the cell. Composition of plasma membrane. Chemically, plasma membrane consists of protein 60 to 80%, lipids 20 to 40%, and small quantity of carbohydrates. Structure of plasma membrane. Fluid mosaic model of plasma membrane. The model proposes that membrane is a phospholipid bilayer in which protein molecules are either partially or wholly embedded. The proteins are scattered throughout the membrane in an irregular pattern, just like a large iceberg float in the sea. The pattern of distribution of protein can vary from membrane to membrane and also vary on both surface of membrane. The membrane is about 7 nanometer thick. The lipid part of plasma membrane consists of two layers of phospholipids which are arranged in such a way that their hydrophobic ends face each other, while hydrophobic ends are appeared on the surface. The steroids cholesterol are wedged into the phospholipid bilayer at some intervals. The plasma membrane is asymmetrical as their two surfaces and halves are not identical. In general, most membrane protein drift sideways in the fluid bilayer. The proteins within a membrane determine most of the function. Carbohydrates are either attached to proteins or lipid, generally on the outer side of the membrane. Filaments of the cytoskeleton are also present on the inner surface of the membrane. These support the plasma membrane. The lipid part of the plasma membrane controls the fluidity of the membrane. When the concentration of unsaturated fatty acid in phospholipids become greater, the bilayer become more fluid-like. That makes cell membrane more flexible. The cholesterol helps to stabilize the lipid bilayer. It also restricts entry and exit of polar molecules and ions. Function of plasma protein. Plasma membrane proteins. A great variety of protein are found in plasma membrane which may act as transport channel or carrier enzymes receptors, or as antigens. Certain plasma membrane proteins are involved in the passage of molecules through the membrane. Some of those have a channel through which a substance simply can move across the membrane. Other are carrier that combine with a substance and help it to move across the membrane. Some plasma membrane proteins have enzymatic function. Some proteins in the plasma membrane are receptors that receive signal from other cells. For example, hormones circulate in the blood but bind to specific target cells with specific receptors. Some proteins are antigens which enable the cell to recognize other cells. Role of glycolipid and glycoprotein as cell surface makers. Mostly glycolipids and glycoproteins act as cell surface makers. They are involved in cell to cell recognition, and sticking the correct cell together in tissue. Regulation of cells interaction with its environment by plasma membrane. Plasma membrane regulates cell interaction with its environment by controlling the transport of material across the cell. Transport across the plasma membrane occur to maintain a stable Hp and ionic concentration within the cell for enzyme activity. The technique used in studying cell membrane. The biophysical imaging techniques such as atomic force microscopy and super resolution fluorescent microscopy has been developed to study biological structures. Scanning electron microscopy is one of the fundamental techniques for the membrane characterization. It gives the microscopy and topography data of prepared membranes. Scanning electron microscopy can be used to determine the pure pore size in case of porous membrane.